Okay, welcome back. Second half of chapter 6, and I'm going to focus now on mechanisms by which molecules can move across membranes and get in and out of cells. So you may be familiar with this term that selectively permeable. We say membranes are selectively permeable, which just means some things can cross the membrane and some things can't. You may also be familiar with these two terms, passive transport versus active transport. Passive just meaning that there's no outside energy requirement for that transport to happen. Don't need to use ATP or anything like that. Whereas in active transport, you do. We'll talk about that distinction. And so anytime you're talking about things moving around either across membranes or within cells, we have to mention diffusion, which I know you've learned about before. But diffusion is simply defined as the movement of molecules from areas of higher concentration to areas of lower concentration. In this example, you have some, a couple of drops of food coloring or dye or something are going into this beaker of water, and initially those dye molecules are very concentrated, right where the drop hits the water, but pretty quickly we see the dye molecules spreading out into the available space until at some point we reach equilibrium where they are basically evenly distributed with the water molecules, and there is no concentration gradient anymore at equilibrium. And so diffusion is always passive transport. Like I said, you don't have to put in any cellular energy for this to happen. You do need some energy in the form of heat energy, but as long as there's some heat energy, diffusion is just going to proceed just because of the random movement of molecules. And so there's a few things that can affect the rate of diffusion. The temperature is going to play a big role. The warmer it is, the hotter it is, the more quickly those molecules will be moving around, and the more quickly diffusion should happen. The size of the molecules that are diffusing will affect it as well. So the larger those molecules are, usually the more slowly they're going to move and diffuse. And then the intensity of that concentration gradient that we started with is going to affect the rate as well. So the more intense the gradient is, the more concentrated the molecules are to start with, the more quickly diffusion will proceed. And if we're talking about things being able to diffuse across membranes, then of course the property of the membrane is going to affect the ability of molecules to cross as well. So, what does the composition of those phospholipids look like? Are they saturated or unsaturated? How dense or how fluid is the particular membrane? And we've got these terms, permeable versus impermeable. So we already mentioned semi-permeable. So if we say the membrane is permeable, then molecules would be able to cross. If it's impermeable, they couldn't. And there are some molecules that can cross a membrane just by what I'm going to call simple diffusion, meaning just unadulterated, pure diffusion without any assistance at all. And I'm saying that because we're going to see some examples where molecules do get some assistance diffusing across membranes. And if we think about what kind of molecules could do this, they are going to be small molecules, right? That kind of makes sense. If you want to diffuse straight across a membrane, you would kind of have to wedge yourself between those phospholipids. So you're going to have to be small to do that. But you're also going to have to be hydrophobic. So if you're a hydrophilic molecule, what will happen is, once you peek into that interior of the membrane that's very hydrophobic, right? The fatty acid tails there. If you're a hydrophilic molecule, even if you're small, once you get into that hydrophobic interior of the membrane, you're going to freak out and just turn around and go back the way you came. So you're going to have to be small, and you're going to have to be non-polar, basically non-charged, um, hydrophobic, to be able to just diffuse straight through a membrane. And so typically this is going to be restricted to just molecules that would be maybe things like hormones. Those are small lipids, or other types of small lipids might be able to directly cross the membrane pretty easily. Um, Now, we can't talk about diffusion without talking about osmosis. And so osmosis is defined as the diffusion of water across membranes, right? And so, okay, that's fine, but if you're really paying attention, this might be a little bit confusing, because I was just explaining how if you want to diffuse across a membrane, you need to be nonpolar and hydrophobic, and of course water is obviously not hydrophobic, right? Water likes to interact with water. So how does water even diffuse across membranes? Um, and it can, and as I'll show you in a minute, it sort of cheats. It basically has some help, right? But here's some examples of some small, for the most part, non-polar type molecules that could just diffuse straight across a membrane, right between the phospholipids. And then you've got some examples of molecules that are either too big, like glucose, or too charged, like certain ions, to be able to diffuse unaided across the membrane. Okay, so how does water do it? And like I alluded to, it sort of cheats. And what I mean is there are actually pores or channels um, created by these proteins called aquaporins. It's very aptly named, right? These proteins make pores that let water molecules pass through the membrane. So here's our membrane. You can see the phospholipids. Here's our water molecules. And then these big pink blobs are the aquaporins, and they literally create a little pore, a little channel that those water molecules can now just diffuse straight through. So the aquaporin is basically kind of shielding the water molecules from that very hydrophobic interior of the membrane, so that they can just diffuse straight across. Okay, so that's a little trick that could be used, really for any of these small but non-hydrophobic molecules. We could help them diffuse across with the aid of some of these special pores or channels, and I'll show you some examples of that in a minute. All right, so when we talk about diffusion, um, talk about osmosis, as I said, it is the diffusion of water across that semi-permeable membrane, right, basically through the aquaporin channels. But it is still just diffusion, right? So it's water is diffusing from where it's more highly concentrated to where it's less concentrated. And whenever you see problems or questions that deal with osmosis, it can be sort of counterintuitive, because usually instead of talking about the water concentration of solutions, they usually talk about the concentration of something that's dissolved in the water, right? A solute. So these purple circles here, imagine that's like sugar or salt or some solute that's dissolved in the water. They'll say, oh, you have like a 10% salt solution on the right side of this membrane. Here's our little membrane at the bottom of this tube, and let's say like a 5% salt solution on the left. Which direction is water going to diffuse? And so really I think the easiest way to think about it is, okay, well, if this solution is 10% salt, that would mean it's 90% water, right? We're going to assume there's nothing else in there. So 90% water on the right side, and then on the left, if we have 5% salt, that means we'd have 95% water, right? So where's the higher water concentration? Well, it's on the left. So that water is going to want to diffuse from where it's more highly concentrated to where it's less concentrated. So it's going to move to the right. And there can actually be force associated with this, right? That osmotic force can actually push the water up this tube against the force of gravity. And another layer to this is, of course, if you're thinking of the solute here, um, the solute would like to diffuse as well, right? So it would like to move from where it's more highly concentrated to where it's less concentrated. In this case, it would like to move from right side of the membrane to the left. Um, but keep in mind this is a semi-permeable membrane. So in this case, whatever the solute is, the salt in this case, let's say, those molecules are just too big to be able to cross the membrane, so they're kind of stuck, whereas the water can move. Okay? But theoretically, right, if the membrane was permeable enough, you could have both water and salt diffusing across the membrane, and they would basically be moving in opposite directions, and you would reach equilibrium more quickly in that instance if both molecules could move. Whenever you talk about osmosis, there's a few terms that I need to bring up. And this is really when you're comparing two different solutions on opposite sides of a membrane, or solutions maybe inside or outside of a cell, for example. So there's isotonic solutions, hypertonic, hypotonic. And isotonic would mean you have equal solute concentrations and uh, also equal water concentrations on both sides of the membrane, right? So if I'm going to go back to my image here, right? So in this image on the right, we have isotonic solutions here. We have the same concentration of salt on both sides of the membrane, same concentration of water. I'm going to stay on the slide for a second. Hypertonic, hyper always means more. That's going to be whichever solution of the two you're comparing has more solute, right? So more salt in this case. And that's going to be the solution on the right side. Hypo always means less. So less solute, less salt in this case is going to be the solution on the left. So these terms only apply when you're comparing two things, right? I can't just give you a beaker and say, oh, that's 10% salt. Is it hypo or hypertonic or isotonic? Well, you would have to immediately ask, well, compared to what, right? Okay, if I give you two beakers and say, yeah, one's 5% and one's 10%, then you could start answering that question. And so you may be thinking, okay, so what? Hypertonic, isotonic? Well, it turns out, in your body or in living organisms, right, the osmotic environment is really important to maintaining healthy cells. So, for example, if you take one of your red blood cells and put it in a hypertonic environment, meaning, put it in, basically, the cell is hypertonic. No, sorry, confusing myself. Put it in a hypertonic environment, meaning outside of the cell you have more solute, right? Let's stick with the salt example. More salt outside the cell, say in your blood, right? You've got a high salt level in your blood. Well, what's going to happen now is due to osmosis, right, water is going to flow out of your red blood cells, and they're going to look weird and shriveled up like this. So your red blood cells should look something like this, nice and smooth and round with a little dimple in the middle. So that's obviously not going to be good for the health and functioning of your red blood cells. In plants, if they are put in a hypertonic environment, again, same thing's going to happen, right? Water is going to flow out of the plant cells, and you can see the cell wall here is a dark line, and all the interior contents, basically the cytoplasm of the cell, is all kind of pulled away from the cell wall. It's all shriveled up and clumped together in the middle because the cell is just incredibly dehydrated. So animal cells generally like to be in an isotonic environment. We have the same concentration of solute inside and outside the cell. The red blood cell looks very happy. Plant cells look better now, but they're actually not going to be entirely content until they are in a hypotonic environment. So plant cells, if they're in a situation where there's less solute outside the cell, so the water is is diffusing into the cells, they're very happy because that's going to fill up the vacuoles and create that osmotic pressure, so you don't have that turgor pressure, you don't have a wilted plant. Um, Animal cells aren't going to like that, right? Your red blood cells going to balloon up, take on way too much water. It looks like it's about to explode there. So again, the point I'm trying to get across is maintaining the correct osmotic environment, inside your body, inside your bloodstream is really critical for your health and survival. So it is important for biology and overall physiology. Okay, so we talked about simple diffusion, right? Small nonpolar molecules could just diffuse straight across the membrane. Everything else is either not going to get across, or, as we talked about with water, it's going to need some help. So anytime you have something that's diffusing across a membrane with help, we're going to call that facilitated diffusion, right? And there's a couple of different ways we could facilitate diffusion. We could do that with channel proteins like the aquaporins, right? Look something like this, where we just create this channel, and basically any molecule that fits through the channel can just diffuse straight through. But it's still diffusion because these molecules are moving from area of high concentration, in this case outside the cell, to the inside of the cell. Or we could do this with what are called carrier proteins, which are just a slight variation on this theme. Really, the difference between a carrier and a channel is the channel is just an open pore, so anything that fits can move through. But in a carrier protein, there has to actually be some interaction. Let me jump ahead. I'll come back to this. Jump ahead one slide. So here's a carrier protein. In this case, this is a glucose carrier. But you'll see the way most carriers work is something similar to this, where there is not a channel that goes all the way through the carrier protein. It's partially closed, right? So you may have molecules that fit into the opening here, but to really open this up, we have to have a molecule that actually binds with the carrier protein and then changes its shape. So in this case, glucose fits in there, it binds perfectly, there's the binding, and when it does that, it causes this shape change in the carrier that allows it to pass the rest of the way through. So there may be some other molecule, maybe a water molecule, right, comes in here, but it doesn't have the right shape to bind to that particular carrier, so it's just going to go back out, right? It's not going to be able to get all the way through. So it's a little bit more specific, right? The carriers are more specific than just the open channels. But that being said, I'm going to go back one slide here. There are channels that can be a bit more specific in that they can be gated. So these would be channels that are actually by default kind of in the closed position. So the channel is closed, and to open them we need some specific signaling molecule called the ligand that would actually come in and bind to the channel and change its shape. Okay, so it's sort of something in between the generic channel and the carrier here. But once the channel is open here, now basically anything that fits through the pore can move through. Okay. So just different types of facilitated diffusion. We can facilitate it with channel proteins, gated or ungated, or carrier proteins. And this is in contrast to active transport. So diffusion is always passive transport. We don't have to put in any energy. Molecules are moving from high concentration to low. In active transport, we do have to put in energy, and we are actually going to move molecules against a concentration gradient. So we're going to move them from where there's low concentration to where there's already a high concentration. Okay, so a lot of times these transporters will be called pumps because they are literally pumping molecules to an area where they're already very concentrated. Just like a water pump is just unidirectionally pumping water into an area where there's already water. And we can see this is a proton pump, so it's pumping hydrogen ions, aka protons. we can see that we need some ATP, right? We need that energy source to move these molecules against that concentration gradient. Different types of active transporters. It's always directional, but that could be uniport, moving one molecule in one direction, symport, moving two molecules together in one direction, or antiport, moving two molecules in opposite directions across a membrane. And we can distinguish when we're talking about active transport between what's considered primary active transport or secondary, and it's a little bit of a fine line, but there's tons of examples of these, so I did want to at least throw it out there. So primary active transport is easy to understand. This is going to be some transporter that directly uses ATP energy to move molecules across the membrane. So this particular example is the sodium-potassium pump. It will move 3 sodium ions in one direction across the membrane, and then 2 potassiums in the opposite directions. This would be considered an antiporter. And this one is actually critically important for our physiology, because this is what happens in all of our nerve cells, and it establishes what's called a membrane potential, which is a charge difference between the outside and the inside of our nerve cells. And then by rapidly basically reversing that charge difference, we can send electrical impulses down our nerves, right? And that's how they function. So we've got these pumps moving these ions in opposite directions and setting up this charge difference, but they're moving them against concentration gradients, right? We're building up a big concentration of sodium ions outside the cell, building up a big concentration of potassium ions inside the cell. So active transport, we're directly using ATP for this to happen, so primary active transport. And now sticking with this example, let's bring in a transporter that's utilizing what we call secondary active transport, and this is this glucose pump over here. And so this glucose pump basically only works if you have your primary active transporter, the sodium-potassium pump here, working in the same neighborhood. So this glucose pump is it's active transport because it's moving glucose molecules against the concentration gradient, right, across the membrane. We've already got a bunch of glucose inside the cell, but we're pumping more and more. It doesn't directly use ATP, but it does require some of the sodium ions that were pumped by the sodium-potassium pump, right, which was directly using ATP. So the bottom line is these secondary pumps will only work if they have a primary pump functioning nearby. Okay, so it relies on the secondary relies on the primary, if that makes any sense. So again, a little bit of a subtle distinction, but pretty common in terms of what's happening in your cells. So I mentioned this term already, membrane potential. Anytime you're pumping ions or charged particles across a membrane, you are, especially via active transport, you're going to end up with this charge difference, charge imbalance on opposite sides of the membrane, and that is by definition considered a membrane potential. And as I already discussed, that's critical for nerve function. But there's other examples of that as well. So I'm not going to go through this, but just a little slide to help you with your studying that's comparing some of the differences between simple diffusion versus facilitated diffusion versus active transport. So I do want you to be familiar with the differences between those three things. And hopefully you're already there after having seen some examples. So okay, last thing I wanted to talk about in this chapter is what about really large macromolecules, right? These are going to be things that are just too big to even cross through a channel. You'd have to have some huge, gigantic channel protein to get those across, and just wouldn't be feasible. Um, and basically, really large molecules can only get in and out of cells through endocytosis or exocytosis. So endo meaning in, exo meaning out. So endocytosis is the process of bringing, large molecules, large amounts of material into the cell. And as you can see here, this is really taking advantage of the fluidity of the plasma membrane, right? So you can have this big macromolecule or clump of macromolecules, right? Basically pushing the membrane in, or the membrane folds in of its own accord. And it can fold in so much that it eventually just pinches off around that macromolecule or that material, forming now what would be a vesicle, an endocytotic vesicle inside the cell. And this could then move off and fuse with maybe a lysosome if this was something that needed to be digested, or whatever part of the cell this needed to head to. This vesicle could now go fuse with some other membrane inside the cell. And we can distinguish two different types of endocytosis depending on whether we are endocytosing liquid or solids. So there's what's called phagocytosis, which is quote-unquote cellular eating, so that's taking in solid material, whereas pinocytosis is taking in endocytosing liquid fluid. And last thing I wanted to mention with endocytosis is there is a way to make this more specific through what's called receptor-mediated endocytosis. The cartoon here I was just showing you is just some generic, whatever happens to be outside the cell just gets taken in. But you could imagine situations where the cell only wants to take in very, very specific items. And so the way it'll do this is it'll have regions of the, excuse me, regions of the plasma membrane, tickle in my throat, that are covered with receptors. And so these receptors are just proteins on the surface of the cell that have to be bound by a specific signaling molecule. And basically you will only get this little pit, the infolding of the membrane to happen if that receptor, or if that signaling molecule is bound to the receptor. So by default, there's going to be no endocytosis, and endocytosis is only going to proceed if this specific signaling molecule shows up and binds to these receptors. So they'll call this infolding of the membrane a coated pit, just because it's coated with these receptors, right? So this is a way, again, just to make this whole process more specific. Unless that particular signaling molecule is present, we don't get endocytosis. And then lastly, the opposite of endocytosis is exocytosis. And is literally just a reversal of that procedure. So you would start out with materials inside a vesicle in the cytoplasm of the cell. This might have originated from the Golgi apparatus, for example. That vesicle can then fuse with the plasma membrane, and once that fusion happens, now the inside of the vesicle is exposed to the outside of the cell, and whatever was in is exocytosed, or basically spit out of the cell. And this happens all the time in biology. So, neurotransmitters, digestive enzymes, something like insulin, it's all originating inside the cell, but it has to get out to be able to do its proper job. Okay, that's it for Chapter 6. In Chapter 7, we will be talking about cell signaling and communication.